All right, thanks. I just couldn't help but think in that last talk. I remember when I was a child, our pediatrician always emphasized to us how important it is to have a family dinner every night. So here I am 50 years later, and nothing's changed. So good old-fashioned family values, I guess. All right. So what I want to do is actually tell you something that I know about, and that is heart problems in Jacobson syndrome. Forgive me because there are going to be a good number of pretty technical slides. Don't worry about that. I just want you to kind of get the big picture. Just for starters, I'm curious, how many of you have a child that has some kind of a heart problem? Raise your hand. Nobody? Oh, there we go. So it's about half or so. And of those, how many of your children have required some kind of heart surgery? Raise your hand. Yeah, so a good number of those. So it's a big problem. It's the most common type of birth defect in humans. So it's something I'm very interested in, both clinically but also in terms of research. Here we go. All right, so just like the other aspects, only about half of your children have some kind of a major heart defect. And so what I've been interested in is trying to understand what the genetic basis is, not just for Jacobson syndrome, but how that information might apply to the general population of children that are born with congenital heart disease. And that actual number is somewhere around 1%, so it's a lot. So in terms of Jacobson syndrome and heart defects, like I said, about half have a major heart defect. The particular heart defect that I'm most interested in that I'll tell you about is something called hypoplastic left heart syndrome, which is basically a uniformly fatal heart defect unless you either have a heart transplant or 3 open heart surgeries, but neither one is really a cure. The other thing that's very interesting to me from sort of a scientific standpoint is that many of the most common congenital heart defects that occur in the general population also occur in Jacobson syndrome. So again, what I hope to learn from Jacobson syndrome, I think could help that much broader population. So this is the heart defect shown schematically that I'm most interested in that actually occurs at a higher frequency in Jacobson syndrome than any known genetic disorders. So somewhere between 5 and 10% of all children born with Jacobson syndrome are born with this. And I can tell you, in my experience, it is still the single most common cause of death in people with Jacobson syndrome. So you see a cartoon of a normal heart on the left, and what you see on the right is the classic description of a hypoplastic left heart. So I think the most important thing to appreciate is that left ventricle, which is the main pumping chamber to get blood out to the body, is very, very small because typically the wall itself, the heart tissue, the heart muscle is massively thickened. Going along with that, you'll see that the valves on the left side of the heart are much smaller and don't function normally. Sometimes they're completely blocked off. And also the aorta, which is typically, as shown on the left, the largest vessel in the body to get blood out to the body, is oftentimes very, very small. So basically, the main pumping chamber of the heart that your life depends on doesn't form and it doesn't work in hypoplastic left heart. And this is actually a real picture of a human heart with hypoplastic left heart on the right. So again, I think you can see compared to another schematic drawing on the left, the wall thickness is massively increased and it completely almost obliterates that chamber volume, so there's no way for blood to be pumped out to the body. Now, interestingly, the field is very confusing because no one really understands why or how that happens. And believe it or not, if you read a lot of the literature, people will say that that massively thickened heart chamber wall the thickened wall is due to decreased growth. Now, I don't know about you, but when I look at that, how can you say that's decreased growth when it's actually massively thickened? So what I'm going to tell you is what we're learning about the heart gene from Jacobson syndrome, which I think is helping us to explain this phenomenon. And it may have some very important implications for how we can treat in the future this disease and maybe even prevent. To make things more complicated, one reason why this whole field has been confusing is even within this diagnosis, there are a lot of different versions of it, and so that makes things more complicated when you're trying to understand the mechanism. But I just want to focus really on this sort of classic version where you have this massively thickened wall. So of that 1% or so of all infants that are born with a heart defect, about 3% are born with hypoplastic left heart. For reasons we don't understand, it's a little more common in males. These days, it's usually diagnosed prenatally with ultrasound. You can see that abnormal chamber pretty early on. So in the United States, there are somewhere between 1,000 and 2,000 babies born every year. Again, it is the single most common cause of death for all infants born with congenital heart disease, and it continues to be the single most common cause of death in Jacobson syndrome. In the United States, the direct costs are probably over $1 billion a year, so it's a pretty big problem. This just shows you a cartoon of what our surgeons are able to do. It's not a cure, but effectively what they do is convert the right ventricle to become the systemic ventricle, meaning to do the job that the left ventricle was supposed to do. So what you see there is the right ventricle is actually hooked up to the aorta, and the blood that normally goes back to the heart is redirected to actually be connected up there to the lung arteries. So basically what you're left with is a two-chamber heart doing the work that the four-chamber heart was supposed to do. And the problem is the right ventricle was never meant to do that work, and so this becomes a big problem, as I'll talk to you about in a few minutes. So when we think about what actually causes that massively abnormally thickened heart, there are a few different theories, but what I'm going to tell you about what we're learning from the Jacobson syndrome gene, that is, it's probably a combination of maybe decreased flow coming into that heart, let's say from a problem with one of the valves initially, but also something about that ventricle itself that does not develop. So it's kind of two components, if that makes sense. So in terms of long-term survival, the survival in the first year is maybe about 70% for all children born with hypoplastic left heart syndrome. And in the first year of life, usually they've had the first 2 of those 3 open heart surgeries. So if you make it past the first year, you probably have a pretty good chance of making it into your adult years. However, we do know, and I've seen this with my own patients, that there can be a lot of variability. So the first baby that I ever diagnosed with hypoplastic left heart syndrome when I was in my training is now 22 years old and overall is doing very well. But I've had several other patients die when they were teenagers or even younger because their heart failed for reasons that we don't understand, but I think I'm going to show you in a few minutes that we might be learning more about why that might be. So long-term survival, if you make it past that first year, is pretty good into adult, but then after that, a lot of these adults are going to end up requiring a heart transplant for long-term survival. In terms of the genetics, so the gene in 11q is one of just a few genes now that have been identified. But to this day, in the vast majority of those 1,000 or 2,000 cases born every year, we don't know the specific genetic cause. And there's a lot of work going into trying to find that. Suffice it to say, it's very confusing. So like I showed you earlier, this is a similar kind of slide to show you how we've used patients with different deletions that overlap to try to narrow down the region. And we were able to do that using the database of known genes to narrow it down to a region that only contains about 6 genes. And it turned out the one on top, called ETS1, had already been shown to be involved in a heart development in an organism called the sea squirt, which coincidentally we see on the coast in San Diego, where I live. These are different pictures of the sea squirts. They're very beautiful animals, and they actually have a heart that's like a tube. This is a slide that was published over 100 years ago. So this tube actually contracts, and if I can make this work, I'm told it will now. You can see how this tube actually pumps the equivalent of blood in this animal to get out to the body. That's my best special effects slide of the day. Now, already before we identified this as the gene for human heart defects, there have been some very nice studies showing how this gene is involved in heart development in the sea squirt. So if you see those 2 red cells down in the lower part, those are the 2 cells that eventually develop to become that heart muscle and that heart that I just showed you. The 2 higher up cells are actually similar, but they go on to become the muscle cells that make up the tail of the C-squirt. And again, these researchers have begun to define the genetic pathways that this gene is involved with, and it turns out this gene has a critical role in two fundamental processes of heart development. One is how a heart cell actually becomes a heart cell. Let's say instead of a tail muscle, how does that cell know to become a heart muscle cell? And also, how do those cells know to get to where they need to go to even form a heart? So these are very important fundamental functions that this gene seems to regulate, not only in the C-squirt, but what we think may be now similar in other organisms, including potentially humans. And very recently, there was a patient that was found to have a very complex heart defect, like hypoplastic left heart syndrome, that does not have Jacobson syndrome, but actually has just a mutation in this gene, in the ETS1 gene, which provides further evidence that it is really the ETS1 gene that's the cause of heart defects in Jacobson syndrome. So my research lab works with genetically engineered mice to try to study these processes for how genes cause heart defects. And so one of the first things you want to do is just ask, what cells in the heart is this gene working in? Because it turns out the heart, in order to develop, has a lot of different types of cells. And so this blue, don't worry about the details, it's just illustrating the two populations of cells where ETS1 is being expressed, suggesting that these are the two critical populations of cells that ETS1 has a role in for normal heart development. And those two populations of cells, which I'll tell you a little more about for the rest of my talk, are called the neural crest, which is a population of cells that actually originates in the developing central nervous system of the fetus. And then another population of cells is called the endocardium, which is the population of cells that makes up the inner lining of the heart that turns out, interestingly, to have a critical role for the normal development of the heart ventricles, and I'll come back to that point. So then the question is, can a defect affecting either of these two populations of cells, how can that cause these heart defects, including hypoplastic left heart syndrome? And with that information, can we learn something that might help us to either treat these defects better or maybe even prevent? So this is just a cartoon to show you that there's this incredibly fascinating population of cells called the neural crest, a subset called the cardiac neural crest that starts in that central nervous system of the fetus or the embryo, and you can see those orange circles, those represent cells that somehow know to migrate to specific parts of the heart to help form those structures. So you can imagine, either from a genetic cause or maybe even an environmental cause, a toxin, let's say, that anything that affects that normal migration process then would affect normal heart development and lead to heart defects. So in our mice, what we have been able to show at the very least is, compared to a normal heart on the left, if you delete this gene, we get one type of heart defect that occurs in your children called double outlet right ventricle. So double outlet right ventricle is shown on the right. There's something called a VSD, which is a hole in the heart, or a ventricular septal defect. And you can also see that in addition to that hole, the red aorta, which normally is lined up over the left ventricle, is pushed over, and now it's aligned with the right ventricle. And what we were subsequently able to show is that if you just eliminate the ETS1 gene in those neural crest cells, you get these same heart defects. So that suggests that ETS1 has a critical role in neural crest cells for normal heart development, and at least in causing some of these heart defects. And I'm not going to go through all the details, but it turns out what we have shown, either in the embryos themselves or if we take out these cells and culture them, that these cells have abnormal migration ability to do that journey out to the developing heart. We've confirmed that further by going back to some of our human patients to use the technology that I talked to you about earlier, so the so-called induced pluripotent stem cell technology. I don't show it down here, but we've actually been able to, with my collaborator, reprogram human skin cells to become neural crest cells. And as shown on this next slide, again, don't worry about it, but if you look at the left upper part, those three red lines are normal human controls, and the three blue lines are three different patients with Jacobson syndrome, missing ETS1. And in all three cases, they have decreased cell migration. So paralleling our findings in mice, indicating that this probably is a major part of the mechanism causing at least some of these human heart defects in your children. So more recently, my postdoctoral fellow in the lab has done some additional experiments in mice. And what he's done is he's, just like in your children, they all are genetically different even though they all have a deletion, right? So in mice, the counterpart of that is to study different strains. And what my postdoctoral fellow has found most recently is, in addition to those heart defects that I showed you, there seem to be problems with the actual myocardium, that heart muscle. And just what I'm showing here is at different stages of development in the mice compared to the control on top. I think what you can appreciate, like comparing E and F, that thickness is actually thinner in the deleted mice compared to the wild type control mice. So in a way, that's exactly the opposite of what we see in hypoplastic left heart syndrome, but again, it all depends upon the strain, just like in your children, it all depends upon the rest of their genes as to whether or not they have a heart defect and what type of heart defect. And he's gone on to look at this a little bit more carefully, and if you compare on top control at different stages of development to below, what you can see is that thickened, or the normal heart muscle layer up top, and if you hopefully see this, you see a lot of little red dots, so those are cells that are the endocardial cells that I told you about earlier. And what you see in the mutant is with the thinner myocardium, there are fewer of those endocardial cells as a result of deleting this ETS1 gene. So what we think is that ETS1 is required for endocardial cells to function normally, and when you lose that function in some way, at least in this mouse strain, you get fewer cardiac cells, myocytes, heart muscle cells developing. Okay, and the reason for that we've gone on to show is that they just don't have as much ability to divide. So decreased cardiac myocyte proliferation in that heart muscle in the knockout mice. And one other aspect that's very interesting and has, I think, some very important clinical relevance is, you know, the normal circulation or the blood vessels that supply the heart, those are called the coronary arteries. And if you look above, if you see those white dotted lines, those are the developing coronary arteries in a normal mouse heart. And it turns out in these mice, these same mice that have the ETS1 gene deleted, there looks like there's impaired coronary artery development. We're still studying this, but I can show you, I don't know how well this projects, but on the left, all this red staining is the blood supply in the heart. So these are the coronary arteries and that whole system of blood vessels. And I think you can appreciate, compared to the normal heart on the left, that you see less red signaling, less stain. It's less arborized, less branching on the right compared to the left. And that actually may have some very, very important clinical implications, and I want to tell you a story why that may be. So this is a little boy that I take care of with Jacobson syndrome who has hypoplastic left heart syndrome. He is now about 2 years old, and he's had the first 2 of those 3 heart surgeries that I told you about. Now remember, I told you that the first baby I ever diagnosed with hypoplastic left heart syndrome, not with Jacobson syndrome. She's like 22, 23 years old now. She's overall doing really well. This little guy, already by age 2, is developing heart failure. And we're going to do some studies to try to figure this out. What I think may be happening here is just relating to that slide I just showed you. I think because of the ETS1 gene that he has missing, he may not have normal vasculature or normal coronary arteries supplying his heart, which might be the cause of why he's developed early heart failure. Because we know in older people, people develop heart failure because of other causes of poor coronary artery function, like smoking and high cholesterol, having heart attacks. In this case, this may be a developmental defect that directly affects the development of the coronary vasculature. All right, so I told you, of course, my interest is in hypoplastic left heart, but I haven't shown you anything to prove in an animal model that ETS1 can cause hypoplastic left heart. And again, I can't say for sure, but it all depends on the genetic background, and we just haven't found the right mouse strain yet to give us that. But I do want to share with you some other animal systems that are beginning to help us to understand how that might be. So this is a fruit fly, which turns out to have very powerful relevance to human heart development, believe it or not. This fruit fly actually has a lot of the same genes required for heart development, including ETS1. And if you look at that left upper quadrant, that is a normal fruit fly heart. You just see 2 rows of heart cells, so it's a tube. And interestingly, this is a paper that was published many years ago before I came along. And each one of these other figures is a picture of a fruit fly heart in which they have a mutation in our favorite gene, the ETS1 gene. And what you see here is something I think very interesting, like in between those 2 arrows. Compared to the normal fruit fly heart, what do you see? You see way too many heart cells that effectively are clogging up that tube to prevent it from normally functioning. So that's a fruit fly heart, and I thought that was very interesting because if that's true, that might help us to explain what's going on in the human heart problem. And this is a slide that is actually tissue from a baby in the United States that was born with hypoplastic left heart syndrome and Jacobson syndrome. The family chose not to proceed, so this baby died a few days after birth, but we were able to get heart tissue. And in this case, what we see is exactly what we would have predicted, that all those pink, all that is just a bunch of extra heart cells. So this is tissue from the left ventricle. So it's consistent with our model that loss of the ETS1 gene causes too many heart cells to give you this massively thickened wall that then obliterates the chamber and prevents it from pumping normally. Now I want to share with you some interesting work done by a collaborator of mine at the California Institute of Technology, just a couple hours north of where I live. And her name is Marianne Bronner, has been studying the ETS1 gene, coincidentally, because primarily for its role in neural crest, and in this case in a frog. But she also was interested in studying how ETS1 functions in normal heart development in the frog. So she very graciously shared with me these data before she actually published the paper. So just to orient you, on the left are 2 different parts of a normal frog heart. So on the lower left where it says V, that's the normal ventricle of a frog heart. A frog heart only has 1 ventricle. And up above where it says OFT, that's like the aorta or the outflow tract. So that's a normal frog heart. And I want to just skip over to the far right side. And in this case, what they have done is specifically eliminated the ETS1 gene in the cells that give rise to, eventually, heart tissue in the frog. And I think you can appreciate, compared to the far left, the far right, where it says V, what you see is a massively thickened chamber wall and too many cardiac cells, too many heart cells. And I think that was a very, very powerful observation that's consistent with this whole theme that seems to be arising, that loss of ETS1 causes too many heart cells. The other thing that's not so obvious on this picture is that, remember I told you those endocardial cells are required for normal ventricle development? In this experiment, what they also found was that when you have this massively thickened chamber wall, those endocardial cells are gone. They're missing. So it's consistent with our model that ETS1 may be required for normal endocardial cell function, such that when you eliminate those endocardial cells, you get this very abnormally developed heart wall. And here is a normal, I don't know how well you see this. Oh, I guess this video isn't working. That's okay. These are just normal tadpoles. And if you look at the tadpoles that have had that experiment to eliminate the gene causing that abnormal heart, I don't know if you can appreciate this, but these embryos now are all swollen up. Basically, these are tadpoles that are dying from heart failure, okay? And so that is consistent with showing that ETS1 has a critical role for normal heart development, even in a frog. And her then postdoctoral fellow, who is now a junior faculty member at Georgia Tech, Shui Ni, did one more really elegant experiment, and I think this has some really, really exciting implications for the future of hypoplastic left heart syndrome. What I said, remember, is if you eliminate ETS1 in a population of cells that eventually gives rise to the heart in the frog, you get that ventricle that's massively thickened. So Shui did a very clever experiment in which she did the same thing, but in addition to just eliminating the ETS1 gene in those progenitor heart cells, she grafted normal progenitor heart cells to that same heart. And what she found was that, I don't know if you could see on the far left, that's the abnormal heart. And then if you look right next to that where it says N equals 4-6, she actually now shows that grafting those normal progenitor heart cells prevents that abnormal ventricle, suggesting that there may be some factor or factors that can come back in and rescue what otherwise would have become that abnormal hypoplastic left heart-like ventricle. And for me as a clinician, that's extremely exciting because what it suggests is if we could identify this early enough in pregnancy, there might be the potential to intervene by doing something like that, maybe a little more specifically, to prevent that heart defect from ever occurring. So just to summarize, knockdown, well, let me skip over the first part. Knockdown of ETS1 in the progenitor heart population called the mesoderm causes that hypoplastic left heart-like ventricle and at the same time a loss of the endocardial cells. It can be rescued by grafting normal cardiac progenitor tissue. And again, this suggests that the endocardium has a critical role in regulating the development of that ventricle. And I think, like I mentioned before, it turns out that it's well known that the endocardium is required for normal ventricular development. So it turns out there is a very complex set of interactions of signals that start out in the endocardium to promote normal development of that ventricle. And so what we decided to do is test that a little more rigorously. And the way we tested that in the mouse is we could specifically kill off endocardial cells through some genetic tricks very early in mouse heart development. And so this is about halfway through gestation of a normal mouse heart. You see basically the two ventricles and the two atria. And in the littermate in which we genetically get rid of those endocardial cells, we see a very abnormal heart whose growth is basically completely arrested, and these mice embryos die shortly after. So that is a very robust example of how critical endocardial cells are for normal heart development. And if we go back and look a little more carefully at those frog hearts, even though most of them had that very thickened heart muscle wall, we actually see, if you look on the right, something that looks like it's a little bit in between in a few of these embryos, in a subset. The arrest shown by that arrow on the left, but also in some cases it looks like heart cells are starting to proliferate abnormally. So what we think may be happening is there are two steps in this process. One is initially an arrest of heart development, and that's why you get that very small abnormal heart in the most extreme case when we eliminate those endocardial cells very early. And then there's a second phase where you get this increased proliferation to give you that very massively thickened heart ventricle. So this raises another very interesting question, and that is that abnormally thickened heart ventricle just due to having too many heart cells? If so, that might be something that we can eventually address. Or are those increased heart cells themselves actually abnormal in how they function? And so I think this is my last slide, and I want to tell you about a very interesting company called Novogene, in which they can use the same technologies I told you about to reprogram heart cells derived from Jacobson syndrome patients, for example, with hypoplastic left heart syndrome. And they can actually recreate a heart pumping chamber like shown down below. And then they can test how that heart chamber functions, whether it's from a control, like that, versus heart cells derived from that little boy that I just told you about who already at age 2 is developing heart failure and is probably going to need a heart transplant in the near future. And they can do all the same kinds of studies that we do on our patients to characterize really those heart cells. And again, I think that could have some very important clinical implications Because if those heart cells are normal and function, but there are just too many of them, then it suggests that maybe we can interrupt that process early on, or maybe even after they're born, if we can just shave off some of that extra thickened heart muscle tissue, we might be left with a more normally functioning ventricle. All of that remains to be explored. Just to kind of summarize, we started with a human. We've looked at the literature and learned what's already been published, and we've done our own experiments in a variety of animal model systems. And I think really what this means is you really have to take a multidisciplinary approach to do the hard science, because there's never one perfect experimental system, whether it's humans or fruit flies or mice. But I think what's very exciting to me is already we're beginning to have some insights, not only perhaps for Jacobson syndrome patients, but the more general population. And like I said, when we do a cardiac catheterization procedure on that little boy, we're going to now take a very close look at his coronary vasculature by doing a biopsy. And if we see that, in fact, his coronary vessels are abnormal, it probably means there's no hope that his heart function is going to get better. And rather than suffering the same fate that several of my other patients have, meaning early death, we will list them for a heart transplant very soon. So I think, to me, that's very exciting that already, thanks to the work that we've done on children like yours, we may be able to bring this back to help future patients. And I just want to acknowledge two people in my lab right now, two PhDs who are doing really great work that are responsible for a lot of what I've shown you. Lu Wang on the left is a postdoctoral fellow, and Lizu Lin on the right is a senior staff scientist who, thank God, are able to keep my lab going when I have to go spend all my time taking care of patients at the hospital. So this is a young man in San Diego, not with Jacobson syndrome, but he's one of those that fortunately we were able to get to in time who required a heart transplant. And I asked him if he would be willing to donate his heart to me. So I didn't have time to talk about it, but it turns out his heart muscle also has all those extra cells, even though he does not have a ETS1 gene mutation. So anyway, he's holding his heart, happy to be alive with a new heart transplant. And again, besides present and past members in my lab, there are a number of other people that I've been collaborating with throughout the United States to make this possible. And that's it for now. Thank you.